And on to chapter 5, the chapter on lipids. Now, fats have had a bad rap over the years. People have linked the obesity problem with lipid consumption. But in fact, lipid consumption is crucial to our health. Our brain is composed of fat cells. The supporting cells around our nervous system are highly dependent on lipids. So consumption of lipids is important. But which fat to choose? We will explore that in this chapter.
What are lipids? Well, lipids are fats. They're a large and diverse group of substances. They're distinguished by the fact that they're insoluble in water, sometimes called hydrophobic, which means if you were to pour oil over water, they would not mix, they would layer one on top of the other. Many different forms of lipids occur in the body and in food. In the body, lipids are stored in adipose tissue that protect and insulate organs. The lipids that we consume are divided into 3 categories, triglyceride being the most common. About 95% of the lipids we consume are triglycerides, phospholipids, and sterols. Let's talk about triglycerides first. They're the most abundant form of lipid in our diet in our bodies. They're formed of 3 chains of fatty acids attached to a glycerol backbone. The fatty acids are long chains of carbon, hydrogen, and oxygens. On one end, the methyl or omega end, you have a carbon and 3 hydrogens on the left-hand side here. Now, on the other hand, the acid group, also known as the alpha end, you have a carbon attached with a double bond to an oxygen and a single bond to an oxygen and a hydrogen. These chains can vary on length, the degree of saturation, the location of the double bonds, and the isomeric form. Let me talk about that one at a time. In terms of length, well, you can either have what we call a short chain fatty acid, which means there's very few carbons in between the omega and the alpha end. Or you can have a medium chain, which means you have anywhere from 6 to 10 carbons between the omega end and the alpha end of the fatty acid chain. Or you can have a long fatty acid. Basically just means you have 12 or more carbons connecting the omega end to the alpha end. The tendency is that the longer the chain, the more solid the food is. The shorter the chain, the more liquid the food is. The degree of saturation means this. Saturation is the number of hydrogen bound to the carbon atom, which means here every single carbon is fully attached to hydrogens. There's 2 hydrogens attached to it. But if you were to take one of the bonds, make it a double bond, it would remove 2 of the hydrogens. This one's even less saturated. So you have here a saturated fatty acid because the carbon is attached to a minimum of 2 hydrogens. Here you have a monounsaturated. Mono means 1, so there's only 1 location where there's no 2 hydrogens per carbon. And polyunsaturated because there are many instances where the carbons are not attached to hydrogens. In terms of saturated, it seems that the saturated fatty acid tends to be more solid, whereas the monounsaturated and polyunsaturated fatty acids tend to be more liquid. Typically, a saturated fatty acid will be a nice straight molecule, whereas the unsaturated will have a little bit of a kink in them. If you think back to your chemistry in high school, you know that hydrogen atoms have a positive charge. Well, when 2 positive charges are side by side, they tend to repel each other. Here it gets cancelled out because they repel each other, but they also repel each other, keeping this molecule nice and straight. But since there's nothing happening on this side, it tends to cause a little kink in the molecule. What does that kink do? Well, it causes a liquid because the solids stack up nicely. If you think of a box of matches and you were to put all the matches in the box, they would stack up really nicely. But if you were to break every single match and then try to put them back in the box, it wouldn't quite work because they need a little bit more space. So the straight molecules, the saturated molecules, tend to be solids. The molecules with kink in them, the unsaturated molecules, tend to be liquids. The location of the double bonds can also affect the quality of the fat. You typically start from the omega or the methyl end, that's the end with the carbon and the 3 hydrogens, and then you count the number of carbons. 1, 2, 3. If the double bond is after the 3rd carbon, it's called an omega-3 fatty acid. You always count the location of the first double bond. If the double bond was further down, you would count the carbons. It might be an omega-6 or an omega-9 fatty acid. So in this case, if you count the carbons, you have 6 before the first double bond. So this is an example of an omega-6 fatty acid. Here you have 9. That's the example of an omega-9 fatty acids. Omegas 3 and omega-6, your body cannot produce naturally, so you have to consume them, making them essential fatty acids. The isomeric form refers to where the hydrogen is around double bonds. So if the hydrogens are on the same side, you have what we call a cis form, and it causes a kink in the molecule. But you can rearrange them in the laboratory to have one hydrogen on one side and the other hydrogen on the other side. This fixes the kink in the molecule and causes a nice straight molecule. This arrangement is called transform, a trans fatty acid. Trans fatty acids are made in the laboratory. They're not very common naturally. Okay, let's recap. Fatty acids are classified based on these 4 things. The length, they can be short, medium, or long fatty acids. The shorter, the more liquid. The longer, the more solid typically. The degree of saturation, so the number of hydrogens surrounding the carbons. If it's fully saturated, it's a saturated fatty acid. If there's hydrogens missing, causing a double bond between the carbons, it's an unsaturated. To classify them, you count from the methyl end, the carbon at the beginning of the double bond. So this would be an omega-2 fatty acid, which actually does not exist. Isomeric form refers to where the hydrogens are along a double bond. If they're on the same side, it's cis, and if they are opposite to one another, it'd be a trans fatty acid. So the human body is unable to synthesize fatty acids that have double bonds before the 9th carbon, making omega-3 and omega-6 fatty acids essential, which means you need to consume them. Now, omega-3 fatty acids are less consumed by the North American diet. You find them in flaxseed oil, walnut oil, soy, salmon, tuna, trout, basically fish oils. It's recommended that you take 2 servings of fish per week or 1 gram per day of fish oil supplements to get the adequate amount of omega-3 fatty acids. Omega-3 fatty acids are crucial for proper central nervous system and brain development. Omega-6, the North American diet tend to consume enough because you find it in poultry. Let's move on to phospholipids. Triglycerides, or 3 chains of free fatty acids attached to 1 glycerol backbone. Phospholipids, on the other hand, are 2 free fatty acids attached to a glycerol backbone and a phosphate head. These fatty acids have the same characteristic. They can vary in length from short to long. They can vary in the degree of saturation and their isomeric form. But the phospholipid, here I put the chemical composition here. These would be 2 long-chain saturated fatty acids attached to a glycerol backbone, and this is the phosphate head. What sets them apart is that the fatty acid still does not like water. We call it hydrophobic. It's phobia of water. But the phosphate head is hydrophilic, which means it enjoys being in water, which allows them to be a great component of cell membranes. If you've taken biology before, you'll recognize this drawing. This is a cell membrane. It's a double layer of phospholipids. On the inside, you have the free fatty acids that don't like water, connecting to one another. But on the outside, the phosphate head that enjoys being in water. So you have the watery outside of a cell and the watery inside of a cell. The other role that they play is to transport lipids in the bloodstream. Blood is basically water, it's aqueous. Lipids do not like being in water. So the phospholipids can surround the fatty molecules, have the fatty acids on the inside, because fat and fat like each other, the polar head on the outside, such that it can travel in a watery environment. We call these lipoproteins because there's also proteins scattered throughout them. Another job of phospholipid is to emulsify. Do you remember bile, produced in the liver, stored in the gallbladder, gets released into the duodenum to help break down fats? Well, bile is made up of phospholipids because one side likes water and one side doesn't. Phospholipids are not essential to a diet, but we find them in eggs, liver, soybeans, wheat germs, and peanuts. If we don't consume enough phospholipids, it can be synthesized in the liver. The third category of fats is sterols. Basically, sterols are carbon atoms that form rings. They can be found either from plants, phytosterols, or from animals, cholesterol. I always like putting this image here because cream of rice. Rice is a plant, but it'll advertise it being cholesterol-free, which is funny because the definition of cholesterol is coming from an animal. Cholesterol is also not an essential nutrient. Your body can synthesize some. Sterols can be a component of cell membranes. It helps provide structures to the cell. They're very abundant in brain and nervous tissue, so they're crucial to health. Consuming too much cholesterol can be an issue if your body has some troubles regulating the level of cholesterol in the bloodstream. Sterols are also precursors to other substances, so they're the building blocks of steroid hormones, vitamin D, bile acids. So we consume 3 types of lipids. Triglyceride, which is 3-chain fatty acids attached to a glycerol backbone. 95% of the fats we consume are triglycerides. Once they get into the body, they then can be broken down. Phospholipids, 2 long chains of fatty acids attached to a glycerol backbone and a phosphate head. What's unique about them is that they have a hydrophobic side and a hydrophilic side, so they blend well with water. And the sterols, they're ring-like structures made of carbon.